Livestock farming. Livestock farming, raising animals for use or for pleasure. In this article, the discussion of livestock includes both beef and dairy, cattle, pigs, sheep, goats, horses, mules, asses, buffalo, and camels. The raising of birds commercially for meat or eggs, that is, chickens, turkeys, ducks, geese, and the squabs, is treated separately for 14 information dairy cattle breeds, feeding and managing dairy for discussion of food, valuing processes of meat. In the article meat processing for further discussion of breed and horses, the article horse breed of horses. Efficient and prosperous animal agriculture historical has been marked of a strong, well-developed nation. Such an agriculture permits a nation the storage large quantities of grain and other foodstuff in concentrated formation and utilizes cattle, breed of cattle. Pigs,
Scientists are trying desperately to stop a relentless and elusive serial killer of frogs. It's a microbial fungus that touched off a worldwide amphibian epidemic. Hey everyone, Matt here. For more than 2 decades, a microscopic fungus called Batrachochytrium dendrobatidis has been pushing as many as 200 frog species toward extinction. Now there are 2 big reasons this one little fungus, commonly called BD or the chytrid fungus, is so hard to stop. The first is that it fights back. BD shuts down a frog's immune system. Amphibians can usually launch killer immune responses. Their white blood cells would totally murder BD, were it a more pedestrian fungus. But when white blood cells encounter BD, they don't kill the fungus, they kill themselves. A biochemical on BD's cell wall is likely what tricks the blood cells into committing suicide. Researchers suspect this chemical is a carbohydrate, but they don't yet know for sure.
There are a lot of mysteries about this fungus. The chytrid fungus, as we used to call it, is now the chytrid fungi. Frogs are responding with their immune system against this fungus. It's still not a victory for the frogs, but they are fighting.
That's Karen Lips of the University of Maryland. She and her lab have been studying the chytrid fungus since it was first discovered. Remember how I said there are 2 reasons BD is so deadly? Lips's group is now trying to handle the second. BD is surprisingly diverse. Scientists are learning BD has evolved into so many lineages, it's really not one disease anymore. Coupled with the genetic diversity across the froggy order, this problem is way more complicated than researchers originally thought. But Lips and others are working to understand how different species foster and spread the fungus differently. By combining their work with genetic studies of frogs and the fungus, scientists might be able to identify why some animals are more susceptible and how others have evolved to be more resilient. A new study even suggests that exposing frogs to dead BD can improve their odds of fighting off future infections, sort of like a fungal vaccine. Still, the question remains, can scientists work fast enough, and can frogs fight hard enough to prevent the fungus from eradicating even more species?
For thousands of years, humans have bred horses in much the same gradual way nature does. But soon, they could actually be designed in the lab, along with other livestock, plants, and even humans. Because a new technology has made editing the genome almost as easy as using a word processor. Genes are made of DNA, long strings of 4 chemicals, best known by their initials, A, G, C, and T. Together, they form the basis of all life on Earth. This new genome editing technique is so much faster, easier, and more accurate than anything that's come before that it's creating a new paradigm for biological research. It's called CRISPR, which is short for clustered, regularly interspaced short palindromic repeats. Here's how it works. Scientists program a guide RNA on a protein called Cas9 with the address of the targeted gene. The guide RNA directs the Cas9 protein to cut both DNA strands precisely at the correct spot, like a molecular scalpel. A new section of DNA is added to the cell and edited into the original DNA sequence, which now incorporates the characteristics of both sequences. Biochemist Jennifer Doudna was one of the co-discoverers of the CRISPR technology.
It's going to enable a lot of science to be done that was impossible to do in the past.
The CRISPR technology is so precise that it can actually edit DNA down to a single letter, and multiple edits can be made at once. Changes in a genome that may previously have taken centuries can now be made in just a short time. This game-changing technology is one of the most powerful in the entire history of science, perhaps comparable to the splitting of the atom. But, like that historic event, the development of easy, cheap, and convenient genome editing may also bring a new and sobering responsibility.
One of the roles that we as scientists need to play is to really communicate the power of this technology and how we can be responsible. Do we know enough about the human genome to understand the impact of making changes to it in a developing embryo?
In fact, the human genome has more than 20,000 genes, many of which we do not understand at all. Doudna has been a cautionary voice, but she is also excited about the possibility of solving big problems.
I think most people would feel that genome editing in adults is, or at least might be, for some applications, a very appropriate technology. It might be analogous to taking a pill for treating cancer or some other disease.
Could we begin to treat previously incurable diseases like sickle cell anemia or cystic fibrosis, and do it inexpensively?
I like to call it democratizing technology.
The CRISPR revolution is already happening in research labs worldwide. For better or worse, every life on Earth will soon be affected by our ability to reprogram the software of life.